Welcome. We're so excited to have you guys here. Thank you so much for joining us. My name's Mikaley. I'm the Director of Marketing and Events at Tattered Cover Bookstore. I just first off have to say thank you guys. Thank you so much for joining us. We are just so elated by this continuation of community that we're having, even though it's virtual. We just really can't thank you guys enough for continuing to support Tattered Cover. We love doing these virtual events. We very much prefer doing them in person with you, but these are offering us some really unique opportunities to meet authors from all over the world, talking about subjects that I never would have touched on before, and maybe just interesting you in some new things as well, or some of your old favorites. And we really just want to say thank you from the bottom of our hearts. Your orders are keeping us going right now. All of our online orders, we just really are just overwhelmed and blessed. So thank you guys for continuing to do that. You can order online through tattercover.com. Right now we have our website open from Tuesdays at 9 a.m. to Saturdays at 5 p.m. And the reason for that is so we can spend those 2.5 days catching up on your orders so we can get your books to you as soon as possible. Our phones are still shut down, as is curbside pickup. We know that Denver's stay-at-home order is lifting very soon, so we're trying to come up with a plan and see what our next steps are. But for right now, that's where we stand. And I just want to first off start by saying that we're doing this because art is a survival resource. It's something that's vitally important, but often has few safety nets when crises like this occur. And we're here to affirm that creative work is essential, and we're here because we want to elevate it. It's always going to be our top priority. We have this platform, and so we want to use it. And specifically for authors that have been chosen because we believe that you guys are going to love them, and we love their books, and we think that we have a wonderful community that is supportive and will find these works interesting. And we believe that our communities need books right now as much as independent bookstores need our neighbor's business right now. And we are a community space, and while we cannot be physically together, we're hoping that these events allow us to be virtually together and still come together as a community. So if you're wanting to still come together as a community with us, we have a couple more live streams coming up. We have one tomorrow. That one's going to be on Instagram with Amy Meyerson, who's the author of The Imperfects. That's tomorrow, May 6th at 5 p.m. And then we also have Darcy Gechter, who is the author of Amazon Woman, and she's going to be on Thursday, May 14th at 5 p.m. on YouTube as well. But the author that we have today, I'm very excited for. This is going to be so great. You guys have probably heard him from his other two science books that are very popular, The Universe Within and Your Inner Fish, which Your Inner Fish was chosen by the National Academy of Sciences as the best book in the year 2009. He trained at Columbia, Harvard, and the University of California at Berkeley. Shubin is the Robert Bensley Distinguished Service Professor and Associate Dean of Biological Sciences at University of Chicago. And in 2011, he was elected to the National Academy of Sciences. It's my honor to have Neil join me today, and I'm going to go ahead and invite him on the video chat so we can get started. Here we go. He should be popping up any minute now. There he is. Hello. Welcome, Neil.
Great to be here. Thank you. It's great to be here.
Hi, we're so excited to have you. And just for those of you who don't know, can you just start by giving us a little bit of the summary of the book? And then I know you've got a really awesome presentation for us too.
Sure. Well, first I want to say thank you.
Absolutely.
Thanks all around. Thank you, Tattered Cover, for keeping the arts alive. And thank you, the audience, for supporting one of the great indie bookstores in our country, Tattered Cover Books. So thank you so much. So some assembly required. And a little perspective on the modern day. Some assembly required is about the great transitions in evolution. 4 billion years life has been on this planet, and it has made remarkable changes in the history of our planet. We've seen great transitions where fish evolved to walk on land, where reptiles evolved to fly, where one group of monkeys evolved to think and walk and do all kinds of things and change the earth. These great transitions, when you look at them, seem so utterly impossible. Yet we live at an incredibly exciting time scientifically in so many ways, where we have new tools that allow us to explore the fossil record, new tools that allow us to look at DNA and understand DNA and manipulate it and see how it has evolved over time. And we put all those things together and we have a remarkable view, a new view of the history of life and how life changes over time. And that's what the book's about. And that's what I'll give a short slideshow about. All good?
Great. I'll let you take it away.
Okay.
Let me hop on
Let
out.
me grab the screen here. Let's see. Let me steal the screen and see what we got. All right. Well, we're going to begin with one of the greats. We're going to begin with Lillian Hellman. Lillian Hellman was a famously hard-living playwright, a fabulously creative playwright, who had a remarkably rich life. You could tell just by looking at her what her life was like. She was blacklisted by the House Un-American Activities Committee in the 1950s and really suffered under that greatly. But in writing her autobiography, she came up with a line that I think describes the approach to evolution that underlies my entire book and many of the major transitions we see in the history of life. She said, in describing her own life, she said, "Nothing, of course, ever begins when you think it does." Such a powerful statement in so many ways, because it applies to fossils, it applies to genes, and yes, of course, it applies to Lillian Hellman's life as well. I'll give you an example. If you think that lungs evolved to help animals live on land, that feathers evolved to help animals fly, and invention after invention evolved to make great revolutions, you'd be in very good company. You'd also be entirely wrong. And we've known this for over a century. It's pretty remarkable. I'm going to work that through. But some of the greatest steps we see in this are relating to DNA and how DNA works to build bodies and how it evolves. And I'm going to show you some of the surprises. I'm going to give you with one of these surprises to show that nothing ever begins when you think it does. So DNA, we have DNA in every cell of our bodies. We have about 4 to 5 trillion cells inside of us. And inside of each one of those cells, inside of each nucleus of each cell, is a 6-foot-long strand of DNA. Think about that. A 6-foot-long strand of DNA is rolled, packed, and crumbled in on itself to fit inside each of every one of those 4 to 5 trillion cells. There's so much DNA inside of us that if you were to take all the DNA in our bodies and lie it end to end, it would extend from here almost to the planet Pluto. That's a lot of genetic computing power. And what we've done as scientists over the last 2 decades is sequence so many different genomes. You know, we started with the Human Genome Project, but we now have genomes for so many species, thousands upon thousands of species. We can now sequence a genome in an afternoon for about $1,000. I mean, it's truly remarkable. And when we look at that genome, we find incredible surprises. And let me just give you one surprise. And it relates to a colleague, a professor and neurobiologist, Professor Jason Shepard at the University of Utah. Jason doesn't work on DNA per se, but he works on memories. He's a neurobiologist. And he was interested in studying a gene that's involved in memories in people and mice and other animals. And this gene is called ARC, A-R-C. So he's studying this gene ARC, and the reason why is because mice that have a mutation in this gene ARC lose memories. They can solve a puzzle during the day, but the next day they can't remember their solution at all. Likewise, humans that have mutations in the gene ARC also have a variety of cognitive deficits. So he's studying ARC. And what he does is he takes the protein that ARC's made and he pops it under a microscope. So he's kind of looking at the product of the gene ARC, pops it under the microscope and takes a peek at it. And he looks at this, here it is. Doesn't look too remarkable. Look at the slide there and you see these little blebs in there, right? He looks at those, he says, you know, I've seen these before. Scratches his head. I know where I've seen them before. I've seen them, these little blebs in other things. Looks exactly like the blebs made by the protein, the gene that causes AIDS, HIV, the virus that causes AIDS, HIV. So wait a minute, a memory gene in mice and people looks a lot like a virus, the clumps that are made by a virus, such as HIV, the virus that causes AIDS. So he's at a medical school, so he calls a colleague in the building next door who works on HIV, and invites them over to the lab and shows them the slide and doesn't tell them what's on the slide. Says, "What do you see?" And the HIV expert says, "Oh, of course, I'm looking at HIV." Wrong. ARC, a memory gene. How's that possible? Well, they looked at the structure of ARC and they compared it to the structure of HIV, and it turns out a gene that makes memory in mice and people and other animals is actually a modified virus. It's a repurposed virus. The idea is once they sequenced all these different genes and compared them to among different species, they found that the hypothesis that at some point in the distant past, in one of our distant ancestors, a virus invaded the genome, but that virus, instead of doing damage, was somehow, by some unknown mechanism, repurposed and domesticated, put to new uses to serve as a gene that serves in memory. Why do that? Well, these little clumps that HIV makes, you can see them here with that arrow pointing to it, actually helps the genetic material of the virus move from cell to cell. It's kind of a capsule that protects it. That's exactly what those clumps in ARC are doing. They're protecting the genetic material as it goes from cell to cell as animals make memories. Truly remarkable stuff. In fact, our relationship with viruses is incredibly complicated. When you look at the human genome, it turns out that 8% of our entire genome is ancient viruses that are lying in our genome defunct, dead, no longer active. Our genome is a graveyard for ancient viruses and ancient viral infections. That's really remarkable because when you look at our genome, only 2% of our genome are our own genes. We have 4 times more viral genetic material inside our genome than our own genes. And that's just the tip of the iceberg of surprises. We have so many surprises in the genome, many of which relate to evolution. So a gene that serves in memory was once a virus. Likewise, when we look at some of the genes involved in pregnancy and the placenta, they were once viruses as well. So repurposed viruses make up quite considerable amount of our body, but they're also fuel for evolution. Let's look at another example. For many things, it begins here, conception. Think about this for a second. A fertilized egg, you can see a sperm and an egg there at this moment of conception, is a single cell. But inside that single cell is DNA, an enormous amount of DNA and other proteins, such that over time, that single cell divides and becomes a body with 4 or 5 trillion cells. We call going from that single cell to a body with trillions of cells, we call that bodybuilding. Sorry about the slide. We call that bodybuilding. But think about that. There's so much biology in this slide that how do you go from a single cell to a 4 trillion celled creature like the one on the right is remarkable. What you have is a DNA recipe inside the egg that unfolds over time as the egg develops from egg to adult. And inside that transition, that growth, is a lot of evolution, a lot of stuff that tells us about how bodies evolve. Uh-oh, I lost my, hang on a second, we lost the slides. This never happened before. Let's stop sharing. Let's start this again. Sorry about that. We had a technical difficulty with a crash of my slideshow. So let me go back. Let me turn off the standby, please. All right, let's see what's happening here. All right, get it back. Living in the Zoom age, there you go. Okay, so let's go back to this. This is actually make Zoom looks simple compared to this transition here. But I'm going to take us back to the 1800s where people studied development to understand evolution. And it begins with this gentleman here, Auguste Dumeril, who worked at the time of Darwin. And he was a lucky man. He had an amazing job. He was the keeper of reptiles and amphibians in the Natural History Museum of Paris. Really remarkable because what happened was he was in Paris at a time of discovery where people were bringing species back from all over the world. Dumeril received a package one year, and he received a package of this salamander, 6 of these salamanders. And these were collected in Mexico by an expedition. And they realized that these were very special. Look at this. It's a fully sexually mature adult salamander, several inches long. And you can see it has external gills, and it has a fleshy tail. This is clearly an aquatic adult. The reason why these people gave this to Dumeril is they knew Dumeril might be interested in understanding how fish evolved to walk on land. And they thought, well, here is a creature that seems to be an intermediate between life in water and life on land. So maybe studying this critter, he can understand some of the biology of that. Well, he saved these salamanders, he put them away, he was doing something else. So he put these salamanders in his menagerie, let them go for a period of time. And he came back a few months later and boom. Instead of seeing 1 species in his enclosure, he saw 2. He saw a new kind of salamander that magically appeared out of thin air. No external gills. Look at that one on the bottom. Look at the tail. It's not a swimming tail. It's a very different tail. This is a sexually mature adult that's fully terrestrial. New species. In fact, looking at it, it's like a new genus. It's almost like somebody put a chimpanzee in a cage one year. It came back the next year and found both chimpanzees and gorillas in the same cage. Pretty mind-blowing. But Dumouriez was a very good biologist, and he decided to study how they develop. And he found the answer lies in how these two formed in simple changes of development. That is, simple changes in how you develop from egg to adult can bring about massive changes across the body of the entire animal. So we studied the development. He found, if you look at the top, they develop from eggs. The eggs hatch, you have larvae. The larvae swim around in water. They swim around with external gills and a big fleshy aquatic tail. And they have a mouth that can feed by sucking things in, just like an adult. And normal development has those larvae metamorphosing. And when they metamorphose, they go into the creature on the left, where they lose the external gills, the tail reshapes, and it's a totally different animal. But he found that there was a simple shift, that in his enclosure, through a simple environmental trigger, you can get two different kinds of salamanders. You can get one salamander that forms, as an aquatic animal with fully functional gills and a fleshy tail, and others that are fully terrestrial. What's the take-home message? Simple changes in development, just simple triggers. In this case, actually, it's a simple change in a hormone, a thyroid hormone, can bring about massive changes in animals in a single generation. And he noted this, and this is how you can get very rapid change during evolution all across the entire body. Now, that was an example of how studying development, embryology, going from egg to adult, can really tell us a lot about evolution. But we now live in the age of DNA. And we live in the age of DNA where we can actually manipulate the DNA. We can edit it using a technique called CRISPR-Cas genome editing. That is, we can cut genes out, we can put new genes in, we can even rewrite the genetic code. And this tells us so much about evolution. And I want to give you one example. The example relates to a tiny little creature called Parhyali. It's what's known as an amphipod. And my colleague Nipam Patel in Massachusetts has worked on this. And you see one of these Parhyali in front of you. The head is to the left, the rear end is to the right. And what you see is it has lots of different kinds of legs. Look at that. There are certain legs in the front that look like claws. Then you go a little further back, you find some legs that face backwards. Go further back, you find some legs that face forward. And then at the end, you find some legs that look wispy. Now, the reason why Nipam studied this is because it has lots of legs. And he can understand the genes that are active when those legs are found. Now, this is a little complicated, but stick with me. It's really worth it. What he found was, when you look at this, there's a genetic address inside the body for each leg that forms. So for instance, where that backward-facing leg is, you see the red arrow, you could see that's a gray. That's where one gene known as UBX is active. So in the segment of the body where the gene UBX is active, you get a backward-facing leg. Go further back. In the section of the body where you have forward-facing legs, where the blue arrow is, you have two genes that are active, UBX and another one called ABDA. That area is kind of with a slash sort of thing. So the genetic address of that is two genes being active, both UBX and ABDA. And if you go further back, there's another address for the wispy legs, which is ABDA and ABDB, where makes those wispy legs. So what Nipam Patel did is he decided to tinker with these genetic addresses to see if he can change the legs that develop. So he made a Parhyali that lacks ABDA. So basically, he turned the entire midsection into a gray area that has only UBX activity. And what did he do? Instead of having forward-facing legs that you had in the animal on the top, you now have all backward-facing legs on the bottom. He showed by tweaking the genetic address of gene activity in these animals, he can change which legs develop where. Really remarkably powerful stuff, because this stuff is fuel for evolution. But the story gets even better. Turns out flies have these genes too. You're seeing on top there an embryo of a fly and color-coded where these genes are active in the embryo. And it turns out where those genes are active in the embryo determines what appendages form in each segment. It doesn't stop there. We have these genes as well. If you look at a human embryo or a mouse embryo, we have versions of these genes too. They're active in the colors you see in the embryo in the middle. And guess what they determine? They determine what backbone, what portions of our vertebrae, how our backbone forms, and where our legs and arms are positioned in development. So it's doing a sort of a similar kind of thing as we see in Parhyali. Turns out we can do in mice the same experiments that happen in Parhyali. And let me give you one example. Here's a cute little mouse. And you can see shown in the back there is the rear end of the vertebral column, towards the end of the vertebral column towards the tail. Now we're going to look at this with genetic addresses, just like we did with Parhyali. And if you look at this, you could see where the red arrow is. There's several genes that are turned on. We're looking at one gene called HOX10, another called HOX11. There are 3 addresses here. There's an address in the top there with just HOX10 active. There's an address in the back where just HOX11 is active. And there's an address where the red arrow is, where both HOX10 and HOX11 are active. Turns out that's a genetic address that determines which kind of vertebra forms in which part. Where HOX10 is active, you get lumbar vertebrae in the base of the spine. Where both HOX10 and 11 are active, where my red arrow is, that's where you get sacral vertebrae. Okay. Now, what they did is a similar experiment as that NEPOM did with Parhyali. If you knock out using this gene editing technique, HOX11, you have a mouse that just has HOX10, which should mean that the mouse just develops what kind of vertebra? Lumbar vertebrae. And that's exactly what happens. Where the blue arrow is, you're able to shift to form lumbar vertebrae and compare that to where you have the red arrow where you had sacral vertebrae. So by changing the genetic address of these genes, these so-called HOX genes, you can change what kinds of organs develop in the body. And versions of the same genetic address and the same genetic toolkit that make the bodies of flies and amphipods like Parhyali make our own bodies, doing similar kinds of things. So let's get back to the more general theme. Where did, nothing ever begins when you think it does. Let's look at this transition here, the transition from life in water, fish on top, to life on land, a limbed animal on the bottom. If you think about this transition, you could think, well, it kind of seems impossible. So many different things have to evolve at once, arise at once, that it almost seems like it could never happen. Think about it. It requires lungs, right, for creatures to live on land. It requires a neck. Fish don't have necks, but limbed animals do. It requires a certain kind of flat head with eyes on top. And it requires limbs with fingers and toes and, and wrists and ankles. I mean, the list is impossibly long. But the point here is that nothing ever begins when you think it does. Let's take lungs, for example. Here's a lungfish. This is arguably one of the cutest fish around. It's a Australian lungfish. Look at that. You just want to give it a little kiss. But it's a gorgeous little fish. They actually lived a long time. They lived like 84 years. There's one in Chicago that lived to at least 80 years. It was collected when it was an adult. But anyway, so these fish are alive today. There are 3 genera of them. In fact, there are lots of species with lungs, but these, you can look at their lungs. What they do is they have lungs and gills. What they do is they go to the surface and breathe when the oxygen content of the water gets really low. They use their lungs when the water has lots of oxygen. When the oxygen content of the water drops, they go to the surface and use their lungs and breathe. But here's the key thing. They have real lungs. They have lungs that are bilobed like ours. They have lungs with all the features, alveoli, all kinds of things. In fact, the genes that form the lungs in creatures like lungfish are the same ones that form our own lungs. And lungs are not only seen in lungfish, they're seen in all kinds of other fish. And the central point is, the more we look, the more we find that lungs aren't some trait that arose as animals walked on land. Lungs arose eons before fish ever took their first steps on land. Lungs arose in fish trying to live in water that had variable oxygen content. And in fact, when we look even broader, we could see that other aspects of this change are also present in fish well before they ever walked on land. That is, when we discover fossils right at the cusp of the transition from life in water to life on land, like this creature here, Tiktaalik rosea, what do we see? Well, it had fins, but it had inside those fins, it had arm bones with an upper arm, forearm, even parts of a wrist. This is a fish that can do a push-up. This fish had a neck. This fish had a flat head and eyes on top. What does this mean? This means that almost every single trait animals used to walk on land originally arose in fish living in aquatic ecosystems. It wasn't like fish had to wait for mutations and wait for the invention of new organs to walk on land. No, they already existed in fish, using them to live in water. And what that meant is when the opportunity came to walk on land, they are already ready to go. They were good to go. And that story is true with almost every other great transition. If you look at birds, they have feathers, they have hollow bones, they have high metabolisms. The long list of features that birds use to fly and live in their ecosystems is enormous, ranging from respiration to excretion to the skeleton to the feathers itself. But guess what? When we look at the evolutionary record, we find all those traits originally arose in dinosaurs, not in dinosaurs that were flying, but in dinosaurs that were fast running, using those traits to capture prey. Again, nothing ever begins when you think it does. And the reason for this is what happens is evolution is one of the greatest repurposers in the history of the world, because what happens is features arise in one context and then they're repurposed to do entirely new things. Great transitions in the history of life don't always require new features, just use old features in new ways. What does this mean for us? It means we're deeply connected to the rest of life on our planet. If you look at the Nobel Prizes that have gone to medicine and physiology in the past 50 years, they've gone to people working on other species, not humans. They've gone to people working on flies, worms, mice, and in some cases even fish. I like to think that as we discover cures to everything that ails us, that the breakthroughs that will extend and enrich our lives will be based on other animals, such as flies, worms, and even fish. I can't imagine a more powerful or more beautiful statement about the importance of evolution than that. Thank you very much. I look forward to your questions.
That was awesome.
We had a little technical glitch in there, but we survived.
That's okay. It happens. What's really
I've been
great
zooming all day and the slides haven't crashed. So there you go.
That's okay. First time for everything. But so we're going to be facilitating questions. If you have any questions for Neil, then you can leave them in the chat. I'm going to kick it off by one of the ones that we got on one of the ones we got on social media, which is kind of a pivot from what you talked about. But people are very curious about what you're reading right now and maybe what other media or information that you're consuming during this crisis.
What information am I consuming during the crisis?
What are you reading? What are you enjoying? What media are what?
Yeah, I can't say I'm enjoying, but what I'm reading is I'm on the Twitter feed. I don't use Twitter a lot, except I'm on the Twitter feed for a lot of great epidemiologists and virologists. So I'm following their feeds and getting their commentary. So what's happening in science now, and this is really important for the general public to think about, is what you're seeing is science unfolding at a rapid pace in real time. The kinds of discoveries we're used to seeing happening over months and years are happening now over days and weeks. So that time frame is compressed. So as a general public, we're seeing kind of the nitty gritty of the dirty laundry of how science works. It's not the dirty laundry, it's kind of the fuel of how we work. What we do is we collect data, we make hypotheses. A high fraction of those initially are going to be wrong, but we follow the data. We try to say, okay, what's the evidence we need to separate, to distinguish between these hypotheses? And so people are seeing that right now. And I think there's a lot of confusion, right? Because you're seeing science happening in real time. You're seeing one study that says something and then another study that says something entirely else, something entirely different. But that's how science works, right? We work collecting data, making the best hypotheses we have at the time. And we realized to be able to distinguish these, we maybe need to collect better or new data. And that's exactly the frame we're in now. So I follow a lot of scientists on Twitter, quite a few of them, which are really good. I think, this is a time period when we're seeing the rise of the great science journalists. That is the science journalists who are on the sort of coronavirus beat are just truly stunning. Carl Zimmer at the New York Times, Amy Maxman at Nature, Ed Young for Atlantic. I mean, I could go on and on. Laurel Helmuth at the Washington Post. There are some truly remarkable journalists, and these people are doing just an incredible effort. And they're putting out 3 or 4 articles a week of such high quality, and they're tempered, very tempered. So if you want to really get a balanced view, read the great science journalists, and you'll see them in the Washington Post, New York Times, Atlantic, and elsewhere.
So someone wants to know, where does your research for a book begin? Where do you start when you get an idea for this book or for some of your other ones? And how does that process start for you?
The process for writing a book starts with me what's the story I want to tell? It begins that way, about science. What's the science I want to get across? And then what's the narrative path I want to get there? And so for me, particularly in this book, in Some Assembly Required, it was telling the history of science as much as telling the science itself. That is, the research for me is really, I'm fortunate to be in a university with an amazing library, with an amazing bunch of colleagues just right across the quad here. So when I begin thinking about a book, I immerse myself in the library, actually physically in the library with books, not online. There's a difference for me. It just kindles so much inside. And I'll call colleagues and think about it, and I'll chart out, I'll almost do a storyboard for how I want to tell the story for each chapter. And often each chapter, particularly in this book, begins with people working in the 17 and 1800s, because I'm tracing, it's about, we do a lot of DNA in modern molecular biology, but the antecedents for the ideas that we use go back deep in time to, before Darwin. So I study those. I go to historical texts and learn about these people. Sometimes I have to travel to other libraries or do interviews with people who are expert in aspects of the history. And then I just sit down and write, and I just love telling the stories of science. And one of the great joys for me in telling stories like in this book was finding people who've been overlooked, whose work has been overlooked, who had overcome hardship, but yet discovered great things, and oftentimes had to fail and learn from failures, and suffered for their science quite dramatically. And you'll notice in this book a number of the heroes are actually heroines. There are a number of women who worked in the 1800s in a time when women didn't do science in the 1800s, and telling their stories was a great joy for me.
So we have one question from the chat here, and it says, what's the typical time scale of a virus becoming a part of the genome, as your example in the ARC protein? Also, what is the current hypothesis on the mechanism of how this occurs?
Yeah, so there's two questions. One I can answer, one I can't, and that's because science can answer. Science can.
Great.
So the first one is they can invade the genome, within a few generations. Really, what it takes is the virus entering the cell, entering the genome, and becoming incorporated into the DNA, and that can happen in a single generation. But something here has to happen very special. It's not like the virus enters just a skin cell, because my skin cells aren't passed to the next generation. The virus has to enter germ cells. That is, has to enter either the sperm or the egg, because it's those that are going to unite and come together and go to the next generation. So there's two steps here. The first is the virus has to invade the genome, but somehow that viral genetic material has to enter the germ cells, and that can take some time. How does it get repurposed? How is it domesticated? There we have some good ideas, but no real data. This Jason Shepard's work is maybe about 3 or 4 years old. The work on the viral repurposed proteins in the placenta is maybe about 7 years old. So stay tuned. We'll know more in about 4 or 5 years, I'm sure. But it's really an open question how that happened.
I'm personally learning so much. I want to just touch back real quick on the heroines and the heroes that you mentioned as the characters in your story. And you said you were able to pull some out that were overlooked. How did you decide on which ones you wanted to pick? Because I'm sure you couldn't include every single historical figure that you ran into, or every single scientist. So whose stories, and how did you decide which ones to emphasize?
What was the science that drove it? I mean, I really wanted to talk about the science. So my goal was science. I wanted to get across certain scientific points. So there are some stories that I would have loved to have told, but they would have been tangential to the science that I wanted to tell. But a good example of one that sort of met both was this woman, Julia Barlow Platt, who, when I read her story, I mean, literally a tear came to my eye. She was, trained in University of Vermont as an undergrad, interested in biology, had a passion for biology, and was really smart apparently. And she decided to go to Harvard in graduate school. So she went there, but after she got there, she was told that women couldn't get PhDs in the sciences at Harvard in the 1800s. So she was not to be deterred. She went to Germany and got a PhD in Germany. She had to teach herself German to do that. That tells you something about her. So she gets the PhD. She comes back to the United States around the turn of the century, and she starts to work at the Marine Biological Laboratory in Woods Hole, Massachusetts, which was a famous place for doing embryology. And she wanted to study embryos at the time. And so she was working there, and she found a supervisor who was incredibly supportive for her. She made a study, and she found that she was looking at the skull development in sharks and salamanders, and she found that the skull development there was weird. It didn't follow what other people expected, that the cells that formed the skull were different from what other people expected. They were coming from not the head area, they were coming from somewhere else in the body. So she wrote a paper on this, and she was widely derided. I mean, people made fun of her. I mean, literally made fun of her, to the point where she was desperate, and she wrote David Starr Jordan, then president of Stanford University, literally, it was hard for me to read, literally begging for a job. She says, I've done good science. I cannot get a job. I will need to leave the field. He never responded. So she left the field of science and she moved to Pacific Grove, California, where she was elected mayor and then saved Monterey Bay as mayor of Pacific Grove. And then just after she died, it was discovered that that cell type that gave rise to the head was a whole new kind of cell that no one thought about before, that was a cell that's unique to all vertebrate animals, which is an important part of their evolution. You know, for me, that was like, yeah, I got to tell that story. And there's a bunch more like it in the book. But, you know, when you find a story like that, it says so much about the human spirit. It says of the power of the human spirit. She was undeterred. Yeah, and what she was able to accomplish in life, you know.
That's incredible. I again, when I read it, I found her to be one of the most prominent figures. I also love the moment when you got to, I'm gonna blank on the scientist's name, but you got to read his application you were a big fan of his.
Oh, Linus Pauling. Yeah, Linus Pauling, Nobel laureate.
Yes.
Yeah, I got to see his application for a fellowship because I'm on a fellowship committee that for the Guggenheim, which is an artistic and scientific creation fellowship. And I found his application, and yeah, he was classic. He failed everything related to physical fitness, but got A's in chemistry and explosives.
And explosives, that's right. I was like, explosives as a class? I don't think that's a homeschooled one during Corona, unfortunately.
No.
So someone else would like to know, what advice do you have for someone who's wanting to write a nonfiction scientific text?
So the first thing that happens if you want to write a nonfiction book, particularly in science, you got to have fire in your belly to tell the story. That is, it begins with really having a passion for the science and a passion for telling the story of whatever science you want to tell. So, because without fire in your belly, it's going to be hard to make it through the project as big as a book. And importantly, I think the idea is to think about, what science do you want to get across and how do you want to get it across? So for science communication, for me at least, it's very important not to use jargon as much as possible. And so if I want to get away from jargon, because as a scientist, I use jargon all the time, and it's really important because jargon has real power to it. It's incredibly precise. When I say a jargony word, which sounds to most people like alphabet soup, to my colleagues, however, it really means something. It has historical meaning, has precise scientific meaning, but that won't cut it in writing for the general public. So you have to think of ways to get points across without the deep jargon, at least for me. And so that means finding analogies, finding other tools to tell the story, and sometimes visuals, things like that. But again, we're a storytelling species. So it all is about the story. It's story. So yes, have the idea of what science you want to tell, but it's all about the stories, because stories are the way we pull facts together. Stories are the way we remember things. Stories pull us in. Stories humanize things in such important ways. They make it's not just cold clinical rationality. It has there's humanity to it when you can tell a story. So think of the science, but then it's all about the stories you want to tell. Think of the narrative.
That's amazing. I don't know, I'm not going to be able to get this word right necessarily, but it says, someone says, Neil, you should tell everyone that you found and described Tiktaalik what is it?
Tiktaalik Razia.
Tiktaalik Razia on Ellesmere, after Mary Dawson and I were there in the Cenozoic, Mac West.
Oh, fun. Oh, awesome. Hi, Mac. So yeah, so we found a creature, one of the first fish to walk on land. I showed it to you earlier, Tiktaalik Razia. I worked up in southern Ellesmere Island, which is up right across from Greenland in the Arctic Circle. I led a team there who worked there for 6 years to find a fish. So we went back 6 years unsuccessfully. Each time we eventually found it. But it's a fish with, which I showed before. It has arms and legs inside the fins. It has lungs and gills. It has a neck. It's an intermediate, and we designed expeditions to find it. And then we went back a few more times to find more Tiktaaliks. We ended up finding 20 Tiktaaliks, 20 of them. So it's not just like one rare intermediate fossil. It's like there's a lot of them. And since then, other people have found them as well. But yeah, it's people have done paleontology up in the Arctic. It's challenging. We were following the footsteps of some greats, including Mary Dawson, who is this wonderful curator from Pittsburgh. Yeah, it's a remarkable community of people, of scientists up there.
That's so cool. I'm sitting here just geeking out because again, as Neil and I were talking beforehand, I'm a theater and English nerd and have, but this science is so fascinating, and it's something that, is having to bring up high school biology for me. But what I loved about reading your book is that, like you said, it told a story. And as somebody, for me, whose whole life revolves around storytelling, it was so well done and easy to follow and so fascinating as well. I want to ask you, and I'd love it if you could share the story about, just again, being the theater nerd that I am, but how did the Lillian Hellman quote enter your life? And what did it feel like when that
Oh,
struck
this is
such a hard chord?
total random. Yeah, this one's just total psychology. I was watching Maltese Falcon,
Yeah.
Dashiell Hammett. I kid you not. I was writing the book and I was watching Maltese Falcon. I went through this Humphrey Bogart phase. I was seeing Dashiell Hammett. I was reading, so I looked up Dashiell Hammett, and he had a long, very tempestuous relationship with Lillian Hellman. I'm like, and it reminded me of Lillian Hellman, and I was reading about her, and I had to get her, and I got her autobiography, and I'm reading the autobiography, and there was the line right in the, talk about contingency and weird passes, like evolution itself, finding the Hellman quote. And I'm reading, I saw this quote, I'm like, that's exactly the point of my book. So it's like in a Lillian Hellman autobiography. So yeah, it was, don't look a gift horse in the mouth.
There you go. We're going to take about 2 more questions or so. Someone would like to know, how did you learn about planning scientific expeditions?
I apprenticed with one of the greats. So I went to graduate school with one of the great field paleontologists in the last century, and he was leading expeditions to Greenland and Arizona, and I just took notes. I went on my first expedition with him, and I was an utter disaster. So I was like a total disaster. I broke more fossils than I found. I didn't really find much. Couldn't camp. I was like, this kid has no chance in the field. And we got back and he said, are you serious about this? I said, yeah, I'm serious. He said, well, you're gonna have to get serious. Okay. I heard the serious several times there. So so I went back with him again, and he was patient with me, and I got better and better, and I decided I really wanted to do it and went all in. And I took notes from him. So when it came time for me to lead, I started leading my own expeditions in the 90s. And believe it or not, that he became one of my field crew members for the Tiktaalik expeditions. And gladly so. It was just a remarkable thing to have him in his later years join me as leader, and I had learned from him. So it's just, yeah, it was great.
That just sounds like a whole wonderful world full of stories in of its own.
Oh,
I love
yeah.
hearing about them. So we're going to end on this question of, what advice or words of comfort do you have to people during this time with the coronavirus, for you as a scientist or you as a teacher? Do you have anything that you want to say to the viewers that are here watching, probably at home?
Yeah, I mean, science is a fundamentally optimistic enterprise. No scientist begins an experiment or an expedition thinking, I'm not going to find anything. No, they're the ones who don't find anything. Science is on this. You have the entire scientific community of the entire world pretty much focused on coronavirus as a way like nothing before. And human ingenuity, human creativity is boundless in these contexts, particularly when there's a sense of urgency. So, yes, my innate personality, nature is to be optimistic, but I am cautiously optimistic that we're gonna do this, get this sooner rather than later. There's too much good science going on. There's science happening, as I said before, in days and weeks that would formerly take months or years. And coronavirus hit at a moment when we have technologies that are unbelievable. The sequence of the virus came out in mid-January. The potential vaccines by end of January, early February. Antiviral therapies for the cytokine storm, which is the more serious version of the disease, are coming online. So I see this, and there's so much activity that I immerse myself in that. Rather than immerse myself in the politics and the sociology and the other things, I immerse myself in the science. And it is so incredibly elevating to see the creativity and the power of the scientific method. So yeah, we'll get it.
Thank you for that. Can I, honest to goodness, just say thank you? I think those are words that a lot of people need to hear. And I think that I can speak for myself and the other people watching live that was wonderfully spoken. And thank you so much for that. What a great note to end on. We are just so excited to welcome Some Assembly Required into the world. I'm sad because I listened to the audiobook, so I don't have a physical copy, which is a great audiobook, by the way. And you guys can order Some Assembly Required by Neil Shubin here at tatteredcover.com. We've got copies in store, and we'll be happy to ship them off for you. Is there any final words you would like to say, Neil?
No, it's really, yes, it's stay well, stay happy, stay focused. We'll make it through this. And thank you for coming out tonight. And thank you, Tattered Cover, for all you do.
Yay. Well, thank you for giving us these stories to share, and thank you for joining us. We're so excited to have you guys on, and we're, like I said, check our events calendar for more upcoming events. We've got events of all kinds, I promise, for every genre, every reader. And, just make sure to continue supporting your local businesses. If you're ordering out, try and order from a local mom and pop restaurant, or, shop locally as best you can. So we appreciate it. And as he said, stay healthy, wash your hands, and we'll see you guys next time. If you could stay on, Neil, we'll just wave bye to everybody. Thank you. And end the stream.
Thank you, everybody. Thank you.